Welcome to Integrative Bioanalytics. Today, we will go over how to define the final cell clusters for single-cell RNA-seq. Cell population identification is a collaborative process. We provide preliminary biological annotation, but the initial clusters and markers are strictly mathematical. Your insight is required to define the final populations based on your research goals and expertise. There are many ways to explore the data, but the steps are simple. Pick a resolution that shows the cell diversity you need. Then check the gene markers and decide which clusters to merge, rename, or throw away. To make it easier, we will walk you through the process with a real example. Muscle samples from healthy mice and a model of muscle wasting. Let's begin with the cluster report. You will see a UMAP for each resolution, the automatic cell type labels we assigned, and the key marker genes. Choose the resolution that identifies all your relevant cell clusters, even if there are redundant populations. We will merge these later. If you have multiple conditions, check the condition UMAP. This will reveal condition-specific populations. Here, for example, 2 populations show up only in the muscle wasting condition. If you open the population folder, you will find bar plots and tables with the exact percentages. Now, let's look at marker genes. We suggest starting with the dot plot. In our example, adipocytes have very distinct markers, but these other 2 clusters look very similar. If we check the cluster tree, they come from a single cluster that split into 2. Write that down. You may want to merge these clusters. After the dot plot, you can check the violin plots, feature plots, and heat maps. By then, you will have a solid idea of each cluster's identity and which ones to merge. You can check even more data. In the attached tables, you have a complete list of markers for each cluster, with fold change expression compared to other cells, percentage of cells expressing the gene, and a marker score. We also provide an interactive data browser. Launch the file matching your operating system and visualize expression of all genes detected. You can also check gene expression in custom cell groups. Our approach is to show you everything, so you can decide what to keep. Take this cluster for instance. It's spread across the UMAP because the main markers are stress and mitochondrial genes. That's a classic sign of cells damaged during tissue dissociation. It should be discarded. Now you are ready to complete the Excel file with the clustering proposal. Just list your final clusters and which numbers they match at your chosen resolution. In our case, we use medium resolution. We will call our first cluster denervated, and we will merge clusters 3 and 10. Cluster 24 becomes the adipocyte cluster, and cluster 18 should be deleted. Do the same for all clusters. Leave the custom colors and root state columns empty for now. You are almost done. Send us an email with your clustering proposal and the resolution chosen. We will reply with updated marker data. Review it and let us know if you want to do any other changes. We can fine-tune the clustering until you are satisfied. Then, we will continue with the rest of the analysis. And that's it. A simple, collaborative process to get your clustering just right.